1 in 230 cats are diagnosed with feline diabetes, but many more likely remain undiagnosed. Feline diabetes is incredibly hard to identify. By the time owners have recognized typical symptoms, cats will have to undergo a lot of stress with tests, including urinalyses and many blood tests, but even then, it may be too late. For these reasons, it is incredibly crucial for there to be an early diagnosis of feline diabetes, as it can be most effectively treated in its earlier stages.
We are RHS-Calgary, and we created GlucoLitter. GlucoLitter provides cat owners with a method to monitor their cat's health, specifically looking at elevated glucose levels in their urine, which is indicative of feline diabetes, among other conditions. The current method for treatment involves a urinalysis, which is difficult to do and oftentimes can stress out the cat, leading to higher levels of glucose in their blood. RHS-Calgary would like to provide an alternative. The way our project works is simple. When a cat urinates in the litter box, the owner can scoop out some of the litter that has been urinated on and spray it using our product. If there is a concerning concentration of glucose present, a color change will be produced. With this, owners can book an appointment with their local veterinarian to seek further diagnosis and potential treatment, which is crucial to allow cats to reach remission before it is too late.
Feline diabetes is becoming an increasingly prevalent issue. Also referred to as diabetes mellitus, it is the inability to produce enough insulin to balance out blood glucose levels. Insulin will latch onto cells in order to signal when they must absorb glucose. Diabetes is often classified as two different branches, type 1 and type 2. Type 1 diabetes is characteristic of high blood glucose concentrations due to a decrease in insulin production. In contrast, type 2 diabetes is characterized by the inability of cells to absorb the sugar from the bloodstream. Factors that put cats especially at risk for developing diabetes include obesity, increasing age, and being physically inactive. According to Cornell University, obese cats are up to 4 times more likely to develop diabetes than ideal weight cats. Hence, with the increase in obesity rates of cats, there has also been an increase in diabetes mellitus, making this life-threatening condition even more common. Common symptoms include increased thirst, excessive urination and appetite, as well as weight loss and lethargy. As such, it can be hard for an owner to identify that their cat may be suffering from diabetes, as symptoms are not always obvious. Unfortunately, if left untreated, the condition can result in ketoacidosis and other diseases, which dramatically reduces a cat's life expectancy. The condition itself is not always easy to diagnose. However, as with any other disease, diagnosis is crucial to allow for remission. Having an early diagnosis is incredibly beneficial in this regard, as it allows cats to be treated before the disease has had a chance to progress. Clinical testing often involves the frequent analysis of elevated glucose levels in both a cat's blood and urine. With this comes a challenge. Most cats will naturally experience a spike in blood glucose levels due to the stress of being at the vet. This is known as stress hyperglycemia.
This is where GlucoLitter comes into play. Our product allows pet owners to see if their cat has elevated levels of glucose in their urine without having to deal with factors such as stress-induced hyperglycemia. That being said, GlucoLitter is not diagnostic. Rather, it makes owners aware that they may need to get their pet checked out. After an official diagnosis, veterinary treatment will be required to manage the illness. This involves insulin injections that are usually administered 2 times daily, though it can vary depending on a cat's particular treatment plan. Furthermore, a cat's diet may be changed. For obese cats, specific dietary therapy is often needed. This entails controlled weight loss. Though there is no particular cure for diabetes in cats, the earlier that a cat is diagnosed, the more likely that it will enter a state of remission and will therefore reach a full recovery. This is crucial as the cat will be able to maintain blood sugar levels without hormone therapy. This is what GlucoLitter aims to do, giving owners an early detection system for high glucose levels and increasing the chance that cats will reach a point of remission and a stable recovery.
The target reaction of our product is as follows. GOX acts as an oxidizing agent and it forms an enzyme substrate specific complex with beta-D-glucose, yielding D-glucono-1,5-lactone and hydrogen peroxide. The hydrogen peroxide will then form a secondary biochemical reaction with a colorimetric assay kit containing a dye xylenol orange to produce a color change from yellow to purple. While we are still in our prototyping stages and have not yet completed the genetic engineering aspect of our project, we have already laid out a detailed plan for its real-world implementation in the form of a consumer-friendly kit. This kit would contain something along the lines of spray bottles, one with the working reagent, one with the enzyme, and a reusable or disposable bag to allow for an isolated environment meant solely for the testing of the litter sample. Both of the listed spray bottles will be stored in a refrigerator, and the reaction itself will take place at room temperature, lengthening the product's expiration, making this an ideal at-home testing kit. Overall, our goal is to design a colorimetric testing kit that operates on urine in most kinds of litter and produces a color change that is easy for consumers to visually detect. In the several weeks preceding the iGEM competition, our wet lab team has been diligently working to further create a sound proof of concept which demonstrates that our product will in fact yield successful results. Through rigorous experimentation thus far, we have determined that our target reaction does produce a visible color change in synthetic urine solutions containing high levels of glucose. This was confirmed to an even greater extent by several controls that we included in our experimental design. Currently, we are working on determining a concentration gradient for GOX in order to make our reaction more effective and efficient. However, we stumbled across some challenges throughout our many experiments, one of which was that our controls changed color when in fact they should not have. For this reason, we came up with a hypothesis stating that the hydrogen peroxide produced is volatile in nature and trace amounts of its vapors contaminated other solutions and reacted with them. As a result of the controls being exposed to H2O2, they in turn also changed color. Since then, we have remodeled our experimental design to avoid further contamination between variables and have so far been producing accurate results. Now, you may be wondering where exactly we plan to take our project in the very near future, given that our proof of concept has already been majorly solidified. Our team's ambition is to assemble our own plasmid and create a part that can be contributed to iGEM using the genome sequence of the GOX enzyme. We found a his-tagged genome sequence of GOX, a part initially fabricated by iGEM Manhattan College in 2017. Due to the fact that it no longer exists on the iGEM database, our team decided to attempt to hopefully re-enter into the database for future use by other iGEM teams once we have redesigned it ourselves. With this, we intend to construct our plasmid by way of ordering the GOX DNA fragment with specific restriction sites, which we will then subclone into the PSBC3 plasmid from iGEM. Our construct will have to be created in two separate components. The fundamental plasmid will be received from iGEM, and the insert is from another plasmid, tailored to our desired endonucleases and based on information found by a previous iGEM team, retrieved from IDT, Integrated DNA Technologies. The process of removing one insert from a construct in order to place it into another is known as subcloning, otherwise known as the transfer of DNA from one vector to gain the ability and functionality to study or express a gene of interest. Throughout our processes, we navigated through many bioinformatics sources, including XPACY, an online bioinformatic resource operated by the Swiss Institute of Bioinformatics, providing us access to over 160 databases and software tools. Expacy enabled us to translate our nucleotide sequence to a polypeptide, otherwise known as a protein or sequence of amino acids. Our design for creating a highly specific plasmid construct begins with sending the sequence of GOX and our required restriction sites, XBA1 and SPE1, to IDT to clone the protein sequence and manufacture into a custom plasmid. The insert that will be a component of the IDT brand plasmid will contain the wild-type GOX sequence with a 6X histag site, a TEV site, and 2 restriction sites. Once we receive the plasmid from IDT, the XBA1 GOX SPE1 insert needs to be cleaved through the use of endonucleases using enzymes corresponding to specific restriction sites. Succeeding this phase, we will take the plasmid from iGEM and the restriction sites will be located. Furthermore, we will then take the insert with the fabricated sticky ends and use a ligase enzyme to connect it to the PSBC3 iGEM plasmid. By doing this step, the final plasmid construct is manufactured. Following the construction of our full plasmid, we will preliminarily have to transform it into DH5 alpha competent E. coli bacterial cells. DH5 alpha cells are genetically optimized for the increased and efficient replication of DNA, and this replication is to be done through a QIA prep mini-prep procedure. Then, another transformation will be completed in BL21 E. coli cells, which are genetically modified and optimized to express high levels of protein. The expression of our target GOX protein will then be induced, and we will grow high amounts of the final bacteria in our lab. These bacteria will then be lysed and purified. The substance will undergo the process of elution in a nickel column, which will separate our desired protein from all other cellular components. To ensure we have done this correctly, we will analyze the column using methods of spectrophotometry, which measures the amount of chemicals in a solution using a light beam that passes through samples.
Initially, we had the idea of creating a litter additive. This would involve implementing our working product directly into litter that cat owners can buy. However, we were made aware of some of the safety concerns surrounding this model. Some individuals voiced their concerns about our proposed product, stating that cat owners may be skeptical to purchase a litter that has biological components directly implemented into it. After careful thought and consideration, we revised our initial model and came up with an external spray that does not expose pets to our product, thus expressing minimal to no safety concerns to humans or the environment. Aside from the safety of our proposed product, our team has also upheld thorough safety considerations in the lab. While completing experiments, laboratory safety has always been and continues to be our #1 priority. We work in a Level 1 biosafety workspace. Even with this, our entire team was required to take a written Workplace Hazardous Materials Information System test prior to being allowed to conduct any physical work in the lab. We were required to learn about hazard classification, cautionary labeling of containers, and about how material safety data sheets are formatted and used. Though our experiments so far did not involve any biologically active substances, we have ensured to use proper sterilization procedures in the lab. For instance, all pipette tips were dispelled into proper biohazard disposal containers, which were then cleaned out by a certified biohazard waste removal company that our high school has signed a contract with. We also ensured to spray down all materials with the 50% ethanol solution when it poses no threats to the materials in the environment or to our experimental results, to keep everything as sterile as possible. Materials that were potentially hazardous through way of inhalation were placed in the fume hood to ensure safe ventilation. All materials and physical experiments were then properly removed from the lab and safely stored in order to avoid exposing younger students in our school to chemical substances. Having taken all of this into consideration, overall, our product is very safe, and we have educated our team on the prospect of safety protocols in the case that issues were to arise.
Moving on to the human practices aspect of GlucoLitter. Our hope in creating this project is to help cat owners provide the best possible care for their pets. We wish to keep pets and pet owners happy and healthy by reducing the stress involved with diagnosing feline diabetes. Using GlucoLitter, we hope to make a positive impact on the world throughout the application of synthetic biology. In terms of values, our team aims to aid as many families with cats as possible. We have achieved this by reaching out to individuals of diverse scopes of expertise and discussing our ideas with them to gain crucial insight and constructive criticism. We talk to pet owners, veterinarians, and even members of our own team that have diabetic cats. All the meaningful feedback we received was used to improve our project, and any suggestions that were given to us were incorporated into all three aspects of our project: HP, dry lab, and wet lab. One of our community members raised a concern that the change of color produced by our reaction can vary depending on the color or type of litter it is sprayed onto. From this, we constructed a new model that was more specific to particular compounds, further reducing any potential risk factors. As previously mentioned, our team has also spent time discussing any uncertainty consumers may have with having a biologically engineered compound near their pet. This helped us recreate our model once more into the form of a spray that can be used independently from the litter box to prevent any potential risks. Beyond just the project, our team wanted to know more about the world of synthetic biology and its applications. A desire and passion to make the world a better place encouraged our team to reach new heights with our project. We were able to foster a collaborative effort alongside the University of Calgary, allowing us to receive feedback and mentorship. Their advice helped us enforce our dedication to improving the diagnosis of feline diabetes. Members of our own team have also struggled with handling diabetic cats, giving us not only a better understanding, but more drive than before. The more understanding we gained on the diagnosing of feline diabetes, the more we were able to see how important finding a solution is. For us, the project is about improving the world around us by solving a simple yet powerful problem. There needs to be a change, and here, with RHS-Calgary, we hope to be the factor that triggers that change.
So many cats are suffering each and every day, and it is our goal to make it possible for cat owners to keep their pets safe, healthy, and happy. By making it possible for there to be early remission and diagnosis of feline diabetes, we can help save several lives. With GlucoLitter, we can provide cat owners with the tools that they need to monitor their pet's health at home in a safe and effective manner. This is GlucoLitter, saving lives one colorful litter box at a time.